Bismillahirrahmanirrahim. Today our topic is glycolysis. In the previous topic we have studied that aerobic respiration is divided into three steps. First one is glycolysis, second one is link reaction, and the third one is Krebs cycle. So here is the first step, that is glycolysis. Glycolysis is actually the splitting of one glucose molecule into two molecules of pyruvic acid is called glycolysis. Glycolysis is also called as EMP pathway. It is called EMP because it was discovered by Emden's mayor of Parnas, due to which he named it after his discovery. Glycolysis occurs in the cytoplasm. In cytoplasm there are two parts. First one is cytosol, and which is about 90% water, and the second one is organomeres, cytoplasmic organomeres. So glycolysis occurs in the cytosol of the cytoplasm. In cytosol there is 90% water, so glucose is soluble in water and converted into the ring form. Steps of glycolysis. Glycolysis consists of two steps. First one is preparatory step and the second one is oxidative step. In preparatory step we split one glucose molecule into glycyl aldehyde 3-phosphate, two molecules of glycyl aldehyde 3-phosphate, in which two ATP molecules are used. Then in oxidative step these two glycyl aldehyde 3-phosphate converted into the two molecules of pyruvic acid, which yield some amount of ATP, which is discussed later. First, there is a preparatory step. In preparatory step, the step 1 is glucose combined with ATP molecule. Under the hexose kinase enzyme, this glucose convert into the glucose 6-phosphate plus ADP molecule. Here 1 ATP is used. And then in the second step, this glucose 6-phosphate converted into the fructose 6-phosphate under isomerase enzyme, because glucose and fructose both are isomer of each other. Glucose 6-phosphate then combined with another molecule of ATP and forming fructose 1,6 bisphosphate plus ADP molecule. Here the second ATP used. As we discussed earlier, in preparatory step the two ATP molecules used. Then fructose 1,6 bisphosphate breakdowns into two molecules of 3 carbon. The first one is glycyl aldehyde 3-phosphate and the second one is dihydroxyacetone phosphate. They both are 3 carbon molecules. The dihydroxyacetone phosphate is unstable substance, so it is converted into the glycyl aldehyde 3-phosphate. The reaction is this. It occurs under the isomerase enzyme. Then the second step is oxidative step. In oxidative step, the glycyl aldehyde 3-phosphate converted into the pyruvic acid, in which the steps involved, which are given below. Glycyl aldehyde 3-phosphate combined with single phosphate and NAD molecule and forming 1,3 bis phosphoglycerate plus NADH2. NADH2 is converted into the ATP molecules in electron transport chain, which is discussed later. In the second step, 1,3 bis phosphoglycerate combines with ADP molecule. Thus, it forms 1 ATP molecule and 3 phosphoglycerate. Here is the carbon number of the molecule. The 3 phosphoglycerate then convert into the 2 phosphoglycerate molecule, that is only a carbon. Here the phosphate attaches to the carbon number 3 and in this molecule the phosphate attaches to carbon number 2. Then 2 phosphoglycerate converts into 1 molecule of pyruvic acid plus forming the phosphoenol pyruvic acid, phosphoenol pyruvate or simply phosphoenol pyruvic acid. which is a 3-carbon molecule. And then the phosphoenol pyruvic acid combined with another molecule of ADP, thus forming the pyruvate, which is the end product, plus also forming one ATP molecule. Here are the products, or simply called as highlights. As discussed earlier, that glycolysis occurs in cytoplasm. In cytoplasm, it occurs in the cytosol, which is the liquid part of cytoplasm, and here the glucose is present in the ring form. It is one of the MCQs which I have faced in the MDCAT. In glycolysis, the starting substance is glucose molecule, while the end product is 2 molecules of pyruvic acid. The gross ATP formation is 4 ATP. Here 4 ATP forms because in preparatory step the one glyceraldehyde 3-phosphate form and the second one is dihydroxyacetone phosphate. So dihydroxyacetone phosphate is unstable substance, so it converts into the glyceraldehyde 3-phosphate. So in oxidative step we used 2 glyceraldehyde 3-phosphate, one above and the second one is below. So all the products which are formed are 2 in number. As the reaction proceeds, so due to which the end product is 2 pyruvic acid. One is this one and the second one is this. The gross ATP formed are 4 ATP molecules. 1, 2, 3 and the second one 4. In preparatory step we used 2 ATP molecules, so net ATP is 2 ATP molecules, and here are 2 NADH forms. One NADH converts into 3 ATP, which occurs in electron transport chain. The NAD molecules 1 and 2 molecules, so 2 NADH converts into 3, so 2 NADH converts into 6 ATP molecules. In glycolysis the net ATP forms are simply called as 8 molecules. Thank you. If you like my work, then please like, subscribe and share my channel with your friends. Thank you.